May we now invite Professor Sharpless and Nielsen on stage to join us in the Q&A session. Hello. Hello. Yes. So can we have any questions for our speakers from the audience, please? Yes.
Good morning. My name is Huang Junye. I'm a student in NTU studying material engineering. I'm very glad to have a chance to ask questions. I have two questions. Both are directed to Professor Sharpless. As we all know, the Nobel Prize is the world's most prestigious ever. As the Nobel Prize laureate, could you please share some experience of the change, such as in your research career after you got the Nobel Prize?
Was the question, does it affect your research career?
Yeah.
I don't know. I try to. I come to things like this because the students are. I'll go to anything that involves teaching, but I won't go to where I stand there and sit there and smile and talk to people. I mean, I really am shy actually, and I don't like to. I like to be alone a lot, and I don't like to be the prize pig, but I do it sometimes. But I'm sort of like Feynman was my goal, my ideal in that, and you know he almost refused the Nobel Prize, and but he found out somebody told him. I learned today that it was going to he was going to get 10 times more problems if he refused it because nobody had ever done that. And he said, "Oh, I better take it then." And also, it didn't change me. I just need to get in deeper. I got to find something that keeps me crazy, excited. Otherwise, I really get. I don't know. I don't know what I might do. I go sailing like Craig. That would be my hope. I get to go sailing around the world or something, if I have time. But other than that, I don't really have. I like dogs a lot, and that's about it.
Oh, thank you. My second question is, I come from China, but China hasn't got many Nobel Prizes. So, Professor Sharpless, what do you think the problem is for China? Do you think it's the creativity of Chinese or the research environment?
China is just doing everything right in terms of infrastructure, and they just turned it around since I was once there in Beijing and I couldn't see. It was winter in November and all the coal was, and people were riding around with fish hanging on the handlebars and they could see, but my wife and I couldn't, and it was really different then. And now China, well, the thing that I worry about is I don't like the idea of publication. I mean, you can do great, if you had a network, you could do great work without formal publications, but you do need to publish a few good papers. But I think people are publishing too much, and just, you can't do hard projects if you write everything up. You got to leave a lot of stuff lying. And of course, you can't do that and have students want to work for you, but some people that come to you maybe are pre-selected that way. Maybe they don't care so much about papers either. And, you know, I think that's the sad part. In China, the thing I see is all this stuff, they don't have all this tradition of drug use. I thought it would be very neat. They could try a new method, like fishing for it or looking for it by letting things flow through organisms. But no, it's something about the tradition and the reverence for nature, and they want to work with the natural herbs and stuff, and that's not necessarily very aggressive. It's kind of, well, it might work well if you had a good way to analyze things. I just hope a lot for China because they're really trying, and the kids are very enthusiastic, you know, but they don't seem to do the risky experiments. That's what I'm afraid of. But so many good ones, so many good papers out of China. I know there's a lot of ones that are Me Tooers, but see a lot of good ones. I think Holland is hard to compete with, though. This little country really does shake things up. You know, they like to get mixed up disciplines. Get rid of the disciplines if you can. Boy, it's so hard, especially in a culture that's thousands of years old. It's going to be harder. The end of science. Where is this thing about the end of science? I mean, it's like end of experts, maybe. And you know what experts are good for? They're good for being in a kind of shell that's not commensurate with anything around them. So there are a bunch of balls rolling around that don't work. That's what I think.
Okay, thank you, Professor Sharpless. So now we'll take a question from our Korea counterparts. Yes, do we have questions for our speakers today?
Hello?
Yes, we can hear you.
My name is Junyoung Kim from Yangjeong High School, and I am interested in biology. I have a question to Professor Jens Nielsen. My question is this. Would it be possible to make yeast live in the human body and continuously make insulin?
So the question is, can we make yeast to produce insulin?
Inside our human body.
Okay, so that would probably not work because we would have an immune response towards the insulin. But there are works going on of course, trying to work on stem cells involved in insulin production. But this is still a lot of hurdles in that direction here. So I think that will still proceed for quite some time in terms of producing pharmaceutical proteins and using those as protein drugs.
Then can we make a mechanism to have the yeast inside the mechanism and release the insulin when it is possible?
So if you could grow the yeast in the human body and then have it to produce the insulin, that's your question, or?
Can we build a mechanism to make yeast grow in it and release insulin when possible?
Yeah, so I think it will not be possible to basically have the yeast cell as an implant, as you could imagine, in the human body because of immune responses and so on.
I understand.
I have a question too. Hi, I'm Ji Song-in from Yangjeong High School. I have been inspired by Professor Nelson's speech. Therefore, I really want to major biology in university. However, I have a question to Professor Nelson. At the beginning of your speech, you said that you have been inspired when you were students. I really want to know what exactly inspired you, and if it is possible, can you give me some advice?
Yes, as I said, early on in my career, I thought that when I was going to graduate as a chemical engineer, that I would work in business or companies. I think what challenged me there was particularly my mentor at that time, who opened up and kind of illustrated to me some of the big questions there are, some of the big problems that you can address using biology and biotechnology. And that inspired me to continue to be involved in research. And I think this is the key point, that you need to find this inner inspiration. You need to find some big questions that you're really interested in solving and contributing to, and that becomes a key driver. And that was basically what happened to me. And I thought in the beginning that I would probably just stay in academia for a few years, but it's pretty obvious that never happened. But on the other hand, with that said, I would also say that if you do an academic career, you also have fantastic opportunities to collaborate. Through my career, I collaborated closely with industry, so I often say that I get the best of both worlds. I have this opportunity to work with young, dedicated researchers all the time, and at the same time learn from industry what are some of the fundamental problems that we can work on a more strategic basis.
Maybe Professor Weiss has something to add to?
About inspiration. I was inspired by my physics teacher in high school, and I think from that on it was quite clear. I was obviously inspired by all my mentors later on, and by my students and postdocs and colleagues. And it's a source of inspiration, and I totally agree. If you have something that you're passionate about and you're willing to risk and put all of your energy into, you can get somewhere. So.
How about Professor Sharpless? Any advice for students looking to seek a career in chemistry or chemical biology?
Just that you should try to mix it up. Chemistry does help connect things, and that's the main. If things are floating around separately, the most dramatic change is to have one link and they've traveled together. If you can get links like that, a few of them, and they can do things that people won't imagine. A steroid and a nucleotide could go together and make an antidepressant, or I mean, anything could happen. You know, it's just the way the system is. What I love is this story about throwing all the enzymes down on the ground, 10,000. Have you ever heard this? And then you go, because everything's connected, they're living under the same roof somehow, but you throw them down, and then a man is not looking, but he crawls around randomly. I guess he's mixed up once, and he ties things together. Well, 10,000, by the time he ties 3 ties on average for each, he'll pick up the whole lot with one hit. They'll all come together. This is not good. This is gridlock in Manhattan. I mean, you know, they're connected, but all through webs of subtlety that they don't know they're connected. You know, otherwise, how could this thing work? So that's why things go wrong. You know, when the silver bullet just doesn't do what it's supposed to do, because no, it's doing a lot of other things. That's what I think.
Okay, thank you. So thank you, Korean. So do we have any other questions from? Yes.
Okay, thank you. Good morning, honored guests. My name is Ning, and I was here yesterday. I'm a junior year bioengineering student in NTU, and I'm also from China. My questions are also towards Professor Nielsen. So my first question is the metabolic engineering model by computer similar in the way that yesterday presented by Dr. Venter, the genomics engineering by computer modeling? Are there any differences?
Yeah, it's sorry that Craig is not here, so I could tease him about this. But I often say that, and I have a commentary written up about this also, that the synthetic biology approach, we are using tools from synthetic biology, but if you're going to go to the strictest sense and build a cell from the bottom, as Craig Venter is working on, you will still need to know what are you going to put in there. Right now, what they have done, which is a fantastic accomplishment, they have basically reconstructed one of the cells that we already know that exist. So that's kind of a proof of principle, you can say. But if you now want to put different parts together, you will have to know which parts to put together. And then it kind of ends in the same thing as we are basically doing. We are just doing retrofitting of the genome, in this case of yeast. And so you can look at a little bit like it. We are re-engineering the process. We are changing the pipes and so on in an existing factory, whereas the pure synthetic biology is completely building the factory from scratch. Now, that building from scratch has the problem because we don't know, as I said, the function of all the parts, and so it has some limitations. I think it's going to take quite some time before that approach will really be functional, and until then, I think we will do re-engineering and retrofitting. But in the long run, this may definitely become very useful.
Okay, thank you. And may I have another question? It's the same towards Professor Nielsen. What are some difficulties in this approach by computer modeling and subsequently genetically modify the microbes for like industry use? For example, before we do the modeling and genetic modification, we need to know the genes like encoding the proteins as well as the metabolism pathway in the cells. So first of all, we need to use maybe like Professor Kaman's method of probing metabolism in the cells to get to know those fundamental knowledges. So what are other difficulties like facing this method?
So of course, when you make a 30 minutes presentation about this, you can make everything sound very easy. The point is just that in many cases, we think, well, we have a really smart strategy and we get surprised. It doesn't work as we were supposed to do. And this is, of course, the process where we are learning something, because the reason why it doesn't work as we were hoping it should do is because the cell has a lot of regulation that we don't know about yet. And so then we begin to understand how that regulation is there. Can we then change that regulation and then basically get on the track that we would like to go? So this is where actually, I think a good example also where you can say mission-oriented research are pretty directed. We want to reach a certain objective, a yeast cell that produces a lot of stuff. But in that process, we actually do a lot of fundamental science because we do understand about how the regulation of metabolism is functioning. And that is really, I think, for me, very stimulating. I would like to get to a certain goal, but actually in the process, be surprised, learn something, dig into that, and then do new contributions.
Okay, thank you for the exciting technology. And actually, for one of our course modules, we use your co-authored book, Metabolic Engineering. Yeah, thank you.
Good morning, speakers. I am Jasper Heng from Tampines Junior College. I want to modify the question our Korean counterparts mentioned to you all. Perhaps instead of using yeast, which may introduce an immunological response, perhaps you'd like to use gut bacteria that actually exists in the human body already. And also, thinking along this line of thought, what are the possibilities of using gut bacteria to introduce certain types of chemicals into the body? Because we learned that yesterday, 10% of metabolites by such gut bacteria actually found in the blood.
Yeah, that is of course a very interesting prospect of actually engineering the gut bacteria to actually contribute with healthy substances. I mean, now that you have a lot of focus on insulin, and of course there is a lot of interest in insulin in connection with diabetes. Now, with that said though, I think we will have to solve the type 2 diabetes really by different ways than just, you know, giving subjects or the patients more insulin, because basically they have a problem about insulin sensitivity. And we have to get to the underlying mechanisms of that, of treating that. So that's one thing. Now, but I think what we can do probably in the future is to have the gut bacteria to basically provide us with different compounds. Actually, we found that in this study on atherosclerosis that the gut bacteria is producing an antioxidant that we could actually recover in the plasma, in the blood of the patients or the subjects, the healthy subjects. That may be actually one of the preventers also of developing atherosclerosis. So in this way, I think one can go that step further and actually in the future have probiotics that are actually also producing compounds that are very healthy for us.
Currently, is anyone actually undertaking this line of thought?
Sorry?
Currently in the scientific world, is anyone actually taking up this new
Yes,
method?
this is it's still a new field, but it's a actually growing field. There are, of course, some potentially ethical considerations also. There is, particularly in Europe, a lot of opposition against the use of GMO foods, because basically if we talk about probiotics, that would go into our diet. So there are a number of barriers to be crossed, but this is a very expanding scientific field.
Okay.
Thank you very much.
Next question.
Hi, I'm Agil from Temasek Polytechnic and a part-time undergrad with University of Western Australia. My first question is to Dr. Nielsen. Are there prospects of designing a meal that can alter our microbiome to a healthy microbiome? And my second question is a general observation. I was measuring the magnetic field in the ambient environment inside a cell culture incubator. We observed that the magnetic field inside a cell culture incubator was very different from that of an ambient environment. And we read papers that the magnetic field influences gene expression, the structure of gene, regulatory proteins, and so on. So shouldn't we ideally incorporate a magnetic field detector in our cell culture incubator so we have a real control of our experiments?
quite honest, I don't really know. That could, you could maybe work in that direction. I think how that interaction is, I should maybe also just say that right now I think we're just scraping the surface of what is happening there, because this whole interaction between the human and a very complex ecosystem. I mean, we have to realize we're talking about millions of and also thousands of different bacteria that are interacting in an ecosystem. And there could definitely be other effects than just pure chemical. I don't know.
Could we design a meal so that we can alter the microbiome? Would the meal, like an Asian diet, alter the microbiome, or a Western diet? I'm pretty sure that you can affect the microbiome by diet, but whether you can consistently change it in a direction is still not unknown, still not known, I would say. So it may well be that we are disposed for a certain microbiome, but it's clearly also, it will depend on what diet. That's why this triangle I was showing, that it's probably determined both by our genotype, as well as probably also what diet we're eating.
Okay, we have time for one last question.
Okay. Hi, I'm Melvin from Republic Polytechnic. Okay, I have a question for Professor Sharpless. Okay, the first question is, will this click chemistry gives a very high possibility to like synthesize the new drug or the safer drug, or synthesize existing drug in a more efficient way?
One of the collections I have, I was collecting, was giving a few talks, but it didn't become necessary. That was the idea that if you have a drug and you have a handle on it, you can reach out in the different directions. So it's nice to have a core that once you have activity, you've got no stumps or ablated hydrocarbons. You have a little group that could become an alkyne or an azide. But don't forget, we found another reaction now that's maybe even more useful than the copper reaction, although the copper reaction will be still your only hope if you want to get a part per trillion bond. But this other reaction is orthogonal too, and it can also connect things. It's the connecting of things that in almost one or two steps that's really important to get speed over the ground and cheap, right? Most medicines have to be manufactured in a separate division that takes on the horrible challenge that the med chemist ignored. And if you don't use any horrible reactions, everybody can do it on their kitchen stove almost, if they have the parts. I envision a world with 1,000 powders, and you take them on your trips. Sometimes it'll be, the Swiss would take the same thing, because they will, but they pay for it, would be purifying, but it would be the same drug, but it keep moving. The drugs could change with the creatures that are trying to catch us, and they could change if they were just modular. The enzyme puts on a phenyl where there was a methyl, and then you back off with one thing, and you're on again. Now, that's the idea. And who was telling HCV yesterday? It was about the mutation. Sometimes people forget about mutations. Oh. It's really fast. And bacteria do it when they're desperate. They lose their control of their replication fork, and they're dying. So all hell breaks loose. Only when they're almost dying. They're damn good. They don't mutate a lot until they're almost out of ATP or really stressed. And then something happens that they've ingeniously discovered. I mean, everything down there is as smart as we are, and smarter even the bacteria are, frankly.
Okay, I have one last question. Okay, as in the click chemistry, we can produce the reaction with a very few reactant to produce a high yield of product. But just now I saw one of the reactions is that it takes 24 hours to get the product, although you're using the catalyst. So is it possible to faster the reaction by improving like the temperature or pressure? Is it possible?
That's something I'm glad you asked, because one thing I started doing, because it was hard for us to get, the reaction doesn't work. Of course, it doesn't work. There are inhibitors, there are circumstances that slow it down. But if you have a system that's pure chemistry, or DNA would be okay if, but because it's stable, but you can't heat proteins above a certain temperature. But if you can heat it, I recommend to heat it as high as you can instantly without destroying your product, and you'll always get a good yield. There's two reasons. The copper hides in these acetylide complexes, and it gets entropy. Heat throws them apart and makes active catalysts. But the other reason is the things that are there, the acetylene and azide aren't infinitely stable. But if there's copper around, there's no prayer for them to show independent chemistry. We tried to make the Weiss show by giving almost no catalyst and letting it have to make it off a tiny piece of copper wire, and it never could win. You take the microwave up to 200 degrees, and you never saw anything but the copper reaction. Otherwise you would see a mixture. But heat it if you don't ever mess around. Always heat it to, say, room temperature, but probably 40 degrees. I mean, that's body temperature. You can do that probably with even sensitive things. But I would start at 60 if I had to choose. Because getting ready, you don't need to worry if you have no heat liability. And frankly, I'm a German by some instinct, because when students come and they say, nothing happened, it didn't work. Okay, what didn't work? What happened? They got the starting materials back. I said, well, you should never tell me that. I don't want to hear that again. Heat it. And if it's not working at room temperature, take it up 50 degrees. Not 5 degrees every day. Just heat it until it goes somewhere. And goo. Char or burn, and then bring it back and say, it's not good.
Okay, sir, thank you.
Okay, we have time for one last question.
Good morning, professors. I'm Cai Xingchen from Tampines JC. Throughout my studies of physics, chemistry, and biology, there's been one question that's bugging me. How is it that lifeless atoms like carbons, hydrogen, can bond together to form living cells, which form living organisms? In short, life.
So the question is, how does life begin? Is that right? It's a very tough question. Maybe Professor wishes to say something.
I don't want to go there.
The answer is 42, but nobody knows what the question was.
Okay.
Actually, I wanted to say something about the insulin. I heard a story from a Davis professor, Bruce Hammack, a couple months ago, and I really was stunned. It's an evolutionary aid disease. I mean, it's always been with us. We're not allowed to run out of gas, and that makes it like, holy cow! You hit this thing in the fair, the gopher pops up, and it's really the most difficult thing next to cancer. But it's actually good. The last person in a boat to die. Well, the women will live longer than the men, other things being equal from starvation. But the last guy to die, or person to die, will be the old diabetic. Because, is that true or not? Because that makes it a sensible evolutionary thing from days when we didn't get much to eat for a month, right? Is that true? Can you just comment on that?
So you say the old guy that doesn't...
Well, no, the diet, something about the way the glucose just goes out.
Yeah. Okay, yeah, so this is...
a little bit dribbles out, right?
So this is this calorie restriction has been proven to improve aging, basically. So
is that the same thing then?
Yeah, it's the same thing. So basically, if you starve your cells constantly, you will just have less oxidative stress in metabolism, and that it's causing a lot of radicals and so on. So it's proven, and it works for yeast, and it works for humans, and it also works for mouse.
So they're reaching. Higher organisms with a big fuel requirement, they're always reaching towards that state because it gave survival value in the old days. Yeah.
And part of the speculation about the whole obesity-related diseases is also because we've changed our lifestyle much faster than we have actually evolved. The evolution has not caught up with the change in lifestyle, so the time constants are just mixed up, and so we are just having much more processed nutrients today compared with how we are actually evolved to handle.
Okay, thank you, all speakers.
Thank you.
So let's join hands and thank our speakers for their wonderful insights.